Previously, we have also discussed that binding of an adhesion molecule to its substrate would not result in cell adhesion unless that adhesion molecule is also attached to the cell's skeleton or the cytoskeleton. Integrants are no different. Let's see how do integrants bind or interact with the cellular skeleton or the cytoskeleton of the cell. Transmembrane linkers, integrators, or integrants, they mediate the interaction between cytoskeleton and ECM. So this is the important thing, that integrants are basically mediating binding of cell's skeleton with the material present outside the cell. We will talk about that material also in much more detail later on. But basically, this is what integrants are doing, linking ECM with the cytoskeleton. Most integrants connect to bundles of actin. We will look at that. I've already mentioned the integrin alpha 6 beta 4, which is found in hemidesmosomes. This integrin is connected to intermediate filaments and not the actin filaments. Integrin binds to its ligand in the matrix. The cytoplasmic tail of the beta subunit binds to several intracellular anchor proteins. So there are proteins present inside the cytoplasm that interact with the cytoplasmic domain of beta subunit, and basically they regulate the function or binding of integrin to its substrate. I have named a few of these proteins here. However, there are quite a few of them, and they play a very important and specific role. We will also talk about some of these proteins later on, but here just let me say a few names. Talin, for example, alpha-actinin and filamin, also vinculin, not mentioned here, but we will talk about that later on. Anchor proteins can bind directly to actin or other anchor proteins such as vinculin, thereby linking integrin to actin filaments in the cellular cortex. So it can be sort of in an arrangement where an anchor protein is binding integrin and another protein is binding that particular anchor protein, so on and so forth. Ultimately, the final anchor protein will bind a cytoskeletal element. These linkages lead to clustering of integrins and formation of focal adhesions between cell and extracellular matrix. So it's sort of a domino effect. Once few of these integrin molecules bind their substrate or their ligand, they start to cluster. Or you remember we once saw lipid rafts, that how certain membrane proteins can cluster. That was one of the ways membrane proteins could cluster. Also, we said that when their conformation changes, they can again cluster due to weak interactions once they're activated. So these integrin molecules, once they're activated, they start to cluster and also get concentrated in specific regions of the plasma membrane. If the cytoplasmic domain of beta subunit is deleted using, for example, recombinant DNA techniques, the integrins still bind the ligand, but they no longer mediate robust adhesion, and they fail to cluster at focal adhesions. So I've mentioned this previously too. The beta subunit plays a very important role. The cytoplasmic domain of beta subunit plays a very important role in recruiting some of these anchor proteins, and those anchor proteins can make the functioning of integrin molecule more robust. So also, I would like to mention that integrins must interact with cytoskeleton to bind cells strongly to matrix. Just like we have talked about cadherins, they require interaction with direct or indirect. Mostly it is indirect interaction with cytoskeletal elements to hold the substrate or the extracellular matrix in a very strong manner. So what we have talked about is that cytoskeletal attachments, number one, help clustering integrins. So a lot of them come together. So the adhesion, it also improves the adhesion and makes it more robust. Number two, it is thought that conformation that allows integrins to bind the ligand tightly is also caused by some of these interactions between integrins and the cytoskeletal elements. So here's basically the schematic. You can see that here is, for example, an collagen molecule which is part of extracellular matrix. Attached to collagen is fibronectin. It is also present in extracellular matrix. And this particular integrin is binding the fibronectin. And of course, as we have said, there are dimers. Here's, for example, the alpha subunit. Alpha subunit basically is responsible for grabbing the extracellular material very, very tightly, whereas the most important role of beta subunit seems to be recruitment of proteins on its cytoplasmic domain. And these anchoring proteins, some of these proteins are anchoring proteins, ultimately hook the beta subunit to cytoskeletal elements. Here, actin is shown. Actin is one of the types of extracellular matrix. So we have seen some of these anchor proteins, some of the roles of these anchor proteins. And now we are going to talk about these anchor proteins in a little bit more detail.